All right, guys, welcome back to our next lesson, which is going to be Macromolecules of Life. So far we've talked about just basic chemistry, things like atoms and molecules and bonding. Then we talked about some properties of water. And now we're going to continue on in that organization of life. So we know that atoms make up molecules and compounds. And then the next step in the organization is macromolecules. Now, really, all macromolecules are just big organic compounds, okay? Now, the word organic, there's actually a whole branch of chemistry known as organic chemistry, where specifically you focus on these compounds. But the key to being organic is that the compounds contain carbon. So that's kind of the essential in an organic compound. Now, I will say that not every compound that contains carbon is necessarily considered organic, because typically in organic compounds we have several carbons bonded to lots of hydrogens. So if you look over here in these molecular pictures, there's carbon here, and then there's technically a carbon at every intersection here. And notice all of the H's and the O's that are around it. Same thing here, we have multiple carbons. Then we have multiple carbons, we have hydrogens, and the same thing applies over here in nucleic acids. So carbon compounds are considered organic compounds when they're bonded to lots of hydrogens, and these are essentially the compounds that are found in living things. So even if it has died, like a piece of dead wood would still be considered organic because it contains those carbons and those hydrogens. So some things that would be included in organic compounds are sugars and fats, the wood, any of these macromolecules that we're going to be talking about in our lesson today. Now, inorganic typically does not contain carbon. Now, notice it says typically an inorganic compound could contain carbon. So over here in the inorganic side, we have carbon dioxide, but remember that that's CO2. So it's not several carbons bonded to multiple hydrogens. So if it's just one carbon atom and it really doesn't have hydrogens bonded to it, we would not necessarily consider that organic. And so your inorganic compounds are not necessarily going to be found in living things. They could be, because we have non-living components as well. But things like oxygen gas, it's something that we need, but it's not a compound of life, okay? Your metals, your salts, those are not considered to be organic compounds. Now, the four most important organic compounds that we're going to talk about today are these macromolecules. Sometimes they're called biomolecules, and macro means big or large. So in terms of molecular size, they are pretty big. We're talking tens, hundreds, and thousands of covalently connected atoms. So it's not like a water molecule where you have 3 atoms. We're talking 20, 40, hundreds, even thousands, and even greater than that atoms bonded together. So in terms of molecular size, these can be huge, okay? Now we have 4 important macromolecule groups. We have our carbs, lipids, proteins, and nucleic acids. And essentially they each have their own specific structure, which is going to give them their own specific function. In terms of structures, 3 of our macromolecules are what we consider a polymer. And the prefix poly means many. And a polymer is any big molecule composed of repeating subunits. This doesn't even have to be a living molecule. Something like a chain necklace, or a general chain, a spider web, plastics, all of these are polymers because they have repeat units. And those repeat units we call monomers. So mono meant one, so a single unit, and then poly means many, so a polymer has many units joined together. And so the key to being a polymer is that it does have these repeat monomers that make up the structure. Now, the only one that's not really considered a polymer is a lipid. It does have components that make it up, and it is a really big molecule, but it doesn't necessarily have a repeat unit. So as we go through the 4 macromolecules, one of the things that we're going to talk about is what is the monomer that makes up that polymer. Now, in order to build the polymer, or in order to break the polymer, we have 2 major types of reactions that could be taking place. So we have dehydration synthesis. Now the word synthesis means to make. So anytime you synthesize something, you're building, you're making, okay? The word dehydration means to lose water. And so essentially in a dehydration synthesis reaction, we are joining those monomers, okay? So dehydration synthesis is meant to build. We're joining those monomers and we're building the polymers. And every time one of those covalent bonds is made, a water molecule is lost. So this dehydration synthesis is going to occur between a hydroxyl group, which is an OH, and a hydrogen atom, which is just an H. And so you put those two together and what you get is water. So every time these monomers are joined together, a water molecule is lost. Now, another name for this reaction is called a condensation reaction. So dehydration synthesis is the same thing as a condensation reaction because we're losing water every time we make that bond. Now, the opposite of that reaction is hydrolysis. And if we break that word down, hydro means water, lysis means to break or burst. So essentially this is water breaks. So this is where we break the bonds between the monomers, and to do that we add a water molecule. So I kind of like to use this sentence to remember it. We lose water to make, and we add water to break. So in dehydration synthesis, we build the polymer, and water molecules are lost every time we make those bonds. And in a hydrolysis, we break the polymer, and every time we do that, water is added. So they're just exactly the opposite. So here's an example of what it could look like. So here's our two monomers. Now, these could be monomers for carbohydrates. These could be monomers for proteins. These could be monomers for nucleic acids. These could also be joining up the parts in a lipid. So when they come together, here's that OH and that hydrogen. They exit, water is lost, and now I'm left with this covalent bond between my two monomers. In order to break that bond, a water molecule is added, and there we have the monomers split apart. So now we're going to get into our 4 macromolecule groups. And the first one that we're going to talk about are your carbohydrates. And so carbs equals sugars equals energy. Okay, if you know anything about carbs, I want you to know that they are sugars that give us energy. Now, a lot of times you'll see that written as quick energy or fast energy. This is kind of our first source for generating ATP. These are easiest for our bodies to burn as fuel. Okay, they're not the only place where we can get energy from, but they are kind of our quickest and fastest and most efficient molecules to use in order to produce energy. So that's why we say quick energy here. Now, they can also be short-term energy storage. So they can store energy, but it's not for super long periods of time. And they can also be structural. And we're going to talk about the types of structures that they make up. So carbs really are our main energy source. A lot of times people want to think that it's proteins, but we really don't want to break down our proteins. If we go back to that reaction of cellular respiration, it's a glucose molecule that we're breaking down in order to make ATP. So really carbs are our main fuel for our cells and for our bodies. Now there are 3 different types, and here we see those prefixes again. We have mono, di, and poly. And so we're going to have simple sugars, double sugars, and really big mini sugars. So we're going to take a look at each one of these individually. So your monosaccharides, saccharide is just a fancy word for sugar. Your monosaccharide is actually the monomer for your carbohydrate. So this is going to be your repeat unit. A monosaccharide is a simple sugar. It's just one carbon ring, okay? And essentially this is going to include sugars like glucose, which is probably the most well-known. It also includes fructose, ribose, galactose, and these are the ones that we use for quick energy, primarily glucose. And this is a formula that you might want to get familiar with if you don't know it already. So I want you to notice this formula. We have 6 carbons per 12 hydrogens per 6 oxygens. And this is the basic ratio for any monosaccharide. It's a 1 to 2 to 1. If you have a sugar like ribose, it's what we call a pentose sugar. It would be C5H10O5. And so these are kind of your basic structures for your monosaccharides. They're just carbon rings, and they typically either have 5 carbons or 6 carbons in them. Now the next form of carbohydrates is a disaccharide, and di means two, so these are going to be your double sugars. So when two monosaccharides are bonded together via dehydration synthesis, what we get are disaccharides. Some examples of these are sucrose, this is just basic table sugar, maltose, which is in malt beverages, and then lactose, which is going to be your sugar that's in milk. And so these do have to be broken down in our diet, but because they're still relatively small, they're another form of quick energy. We just have to break those bonds. For people who are lactose intolerant, they have trouble breaking down this molecule. They don't have the right enzyme to break that molecule down, and it can cause them some issues in their digestive system. And then the third type of carbohydrate that you need to know are the polysaccharides, and poly means many. And when we say many here, we mean many. Like, we're not talking like 3 or 4, we're talking a whole bunch, okay? These are some of the ones that can get up into those hundreds and maybe thousands of atoms bonded together. And so we have really kind of 4 main examples of polysaccharides that we're going to talk about in our next slide. We have starch, glycogen, chitin, and cellulose. Now, these could be short-term energy storage. They could also be structural. So remember that your monos and your disaccharides are kind of your quick energy. Your polysaccharides are going to be more of a storage molecule, and they can also make up structures. And these are the polymers of our carbs. So the monosaccharide is the monomer, the polysaccharide is the polymer. And so there's 4 important polysaccharides that you need to be familiar with. They're all pretty significant when it comes to living things. Now, these first 2 are both going to be your energy storage. Now, the difference is what kind of organisms are they storing energy in. Starch and glycogen are both big, long chains of glucose molecules joined together. And starch is what you find in plants. And most of us have probably heard or know about eating starches. Like potatoes and pastas, they're all full of starch. Fruits can have starch. But essentially, starch is the way that plants store extra sugar. And then glycogen is the way that animals store sugars. So like when we need energy, we can tap into those glycogen resources. You'll find that in your liver and your muscle cells, and we can break them up into those smaller glucoses, which we can then use to make energy. Now, these other 2 are structural polysaccharides. They both make up cell walls. Cellulose is found in plant cell walls, and chitin is found in fungal cell walls. And both of these are just big carbohydrates. Cellulose is the one that we commonly refer to as fiber in our diet. It's hard for us to digest. So like if you need to poop, they say to eat more fiber. And it's because whatever we can't digest gets kind of pushed through our system. And then chitin is in fungal cell walls. This is also what makes up that crunchy exoskeleton in an insect. And so that takes care of our first group. Remember that carbs are sugars that give us energy. Our next group are lipids, and lipids are fats and oils. That also includes waxes and steroids and phospholipids. And this is the group that we said it's not necessarily a polymer. It does have smaller parts that make them up, but it doesn't necessarily have one unit that repeats over and over and over and over again. Now, the one thing that puts all lipids together in a group is that they are hydrophobic. They are nonpolar molecules that do not like water, and so at least some part of that molecule is going to be hydrophobic. And we're going to take a look at 3 examples of lipids. The first one is what we call a triglyceride, and tri means 3. And you can see over here in our picture that our triglycerides have 3 of these structures called fatty acids. And what fatty acids are long chains of carbons surrounded by lots of hydrogens, okay? So every triglyceride has 3 of these fatty acid tails, and then kind of the backbone of the molecule is what we call a glycerol. So these aren't necessarily monomers, but these would be the main parts that make up a triglyceride. They have a glycerol molecule, which is kind of sugar-like, and then they have these 3 fatty acid tails. And these fatty acid tails are nonpolar, and that's what makes them not like water, okay? So your fatty acids could be described as either saturated or unsaturated, and that's what we're going to take a look at next. Now, if it's saturated, what that means is there's no double bonds between the carbons. And so carbon likes to form 4 bonds in order to have a full valence shell. And so if there's no double bonds, you can see that we have hydrogens going all the way around. And this is going to make for a long, straight fatty acid, okay? These are the fats that are solid at room temp because they're more uniform by being straight. Now, an unsaturated fatty acid has at least one or more double bonds between the carbons. And so what happens is, when you have this double bond here, notice there's some hydrogens missing because now there's 1, 2, 3, 4 bonds, so it doesn't need hydrogens down here. Well, what that does is it causes this fatty acid to kink or to bend. And so unsaturated fatty acids are kinky, and what happens is they aren't as uniform. And so your unsaturated fats are actually liquid at room temp. So your saturated fats are your animal fats, things like bacon grease and lard. These are not the healthiest of fats. These are the ones that contribute to heart disease and clog your arteries. And then unsaturated fats are going to be your plant oils, and these are considered to be much healthier. Now, just an FYI, trans fats is maybe a word that you hear in the news. Trans fats are actually taking liquid oils and they bombard them with hydrogen. So they essentially hydrogenate, or they add hydrogens to these liquid oils, and what that does is it makes them solids. So sometimes you'll see like butters, they aren't true butter, but what they are is hydrogenated oils. And our body doesn't process these quite like it does the other fats. And so what happens is it can affect your cholesterol levels. It raises the bad and lowers the good. And so these trans fats are really not very healthy for us because they've been kind of chemically altered and they're not necessarily in a natural state. So you might want to watch out for foods that are made with trans fats. We do need fat in our diet, but it's definitely better to go with those healthier plant oils that are liquid at room temp. They're better for our bodies. All right, our next lipid is called a phospholipid, and this one's kind of easy because the lipid is in the name. But this one's a little bit different, okay? It has two fatty acid tails. So this is the non-polar hydrophobic region of the molecule. But on the other end, it has a phosphate group and a glycerol. And so having this phosphate group up here, it actually makes the head of this molecule polar. So we have a polar region and a non-polar region. And so phospholipids are what we call amphipathic. And what that means is it's kind of dual state. It has a region that is hydrophilic, that is the head region. It actually likes water. And then it has a region that is hydrophobic, and those are the tails, and they don't like water. And this is essentially the basic structure in our cell membranes. So over here, you've got water inside and outside of cells, and so those fatty acids turn to the inside, and this is what creates this double phospholipid layer that makes up our cell membranes. And we'll talk more about that when we get to cells, but just know for now that phospholipids make up the cell membrane. And then our third lipid that we're going to talk about are steroids. Now, steroids are a little bit different. They don't have long fatty acid tails. Instead, they have this four-ring structure. But what makes them a lipid is they also do not like water. They are hydrophobic. And so a few examples are cholesterol. You can find cholesterol in our cell membrane. It helps to stabilize our membrane. Vitamin D, which helps us with calcium and getting calcium out of the blood or out of the digestive system and into the blood. And then we have two examples of steroid hormones, estrogen and testosterone. And so you can find steroids in different forms. We're not necessarily talking about the bad kind that you take that are illegal to get buff. These are some natural steroids that you'll find in living things. All right, next group are your proteins. Proteins are kind of hard to define with one or two words because there are many different kinds. There's many different kinds of proteins and a wide variety of functions. Now, the one thing that holds all proteins together is that they are all polymers made of amino acids. So the monomer for proteins is an amino acid. There are 20 different ones, and essentially the amino acids that make up that protein determine the function. So we have a wide variety of functions. They can be structural. We have proteins like keratin, actin, and myosin, things that make up the structures in our body like bones and muscles and skin. They can be regulators in the case of like hormones or enzymes. They could be transport molecules. When we get to cells and the cell membrane, we're going to talk about different types of proteins that move molecules across the membrane. And then we also have antibodies, which are part of our immune system and help us fight disease. And this is just a short list. There are many more types of proteins and more functions. That's just kind of an overview. So a little bit more about the amino acids. Remember, there's 20 different ones, and every amino acid has the same basic skeleton. It has this amino group, which contains nitrogen. It has a carboxylic acid group, and this is where you get the name amino acid, okay? So it has an amino group, and it has a carboxylic acid group. Now, everyone has a carbon and a hydrogen there. And what's different, the reason we have 20 different amino acids is because there are 20 different things that you could find right here in the amino acid. Okay, this is called our R group, and it varies among the 20 different amino acids. Okay, so every amino acid has these portions, and they're different because there's something different here. Okay, it could just be something like a hydrogen, it could be a sulfur, it really just depends on the amino acid. Now, this is going to give amino acids different properties. And so when we build proteins, we build an amino acid chain. And so it's the properties of those amino acids and the order of those amino acids that's ultimately going to determine the final protein shape. Now, when it comes to protein shapes, there's 4 levels of structure. Now, it starts with what we call a polypeptide. And so we just build a chain of amino acids. It's amino acid bonded to amino acid bonded to amino acid. It's essentially the basic polymer of a protein. Now, what happens is there start to be interactions among these amino acids. There's going to be some hydrophobic interactions. There could be some forming of hydrogen bonds. There could be ionic bonds that are formed. And so what happens is this chain begins to twist or fold as bonds form between those amino acid R groups. And then what happens is now we have new R groups exposed to other R groups. So we get some more bonding, and that chain twists and folds to form what's called the globular structure. And it literally looks like a very ununiform wad, glob, whatever you want to call it. But it's actually highly specific, okay? Every twist and turn, every nook and cranny goes back to that original polypeptide and the sequence of those amino acids. So every little bump and space is going to be important in a protein because, once again, the shape determines everything. Now, the way you get to the quaternary structure is, let's say there were more than one of these chains making up the protein. That's how you would reach that 4th level. But this right here, the tertiary and the quaternary, these are your functional proteins. So as just a polypeptide, it's not really doing its job yet. It's got to twist and fold and glob up before it's actually ready to be a functional working protein. All right, one special protein to mention, we're going to have a whole lesson on this later on, but one special protein to mention are enzymes. They're very important because they regulate our metabolism, and they do that by speeding up chemical reactions. So one thing that's significant about enzymes and really any protein is that when a protein loses its shape, it loses its function. The shape is everything. And so one thing we have to be careful about with our proteins is we want to make sure that our proteins do not denature. And what happens is they lose that very complexly folded shape. And if they unfold and unravel and they don't have their shape anymore, they do not work anymore. And so this is why we have to maintain homeostasis partially, things like temperature and pH, because if those particular conditions become too extreme, our enzymes denature, and that affects all of the chemical reactions that happen in our bodies. All right, our last and final macromolecule are going to be our nucleic acids. And nucleic acids, a lot of times people are familiar with them because they know these molecules, DNA and RNA. These are our main examples of nucleic acids, and they are polymers that are made up of nucleotides. So this right here is our monomer for nucleic acids. Okay, this is one nucleotide right here. Each nucleotide has 3 parts. It has a 5-carbon sugar, which is why it looks like a pentagon. Okay, this 5-carbon sugar could be either ribose or it could be deoxyribose. and that's where we get the names from, deoxyribonucleic acid or ribonucleic acid. So we have a 5-carbon sugar. We have a phosphate group, which is just a phosphorus and some oxygens, and then we have a nitrogen base. And so these nitrogen bases are the pieces of your DNA and your RNA that actually hold the genetic code. We know that the genetic code is in our DNA, and we can copy that into RNA, but really this right here is where the genetic code lies. And so just a quick comparison of the two molecules. In DNA, this is our main blueprint. This is the one that's found in the nucleus, and it holds the instructions for who we are. We have sequences of these nucleotides called genes. Your DNA has 2 strands, and you can see that those 2 strands are held together by hydrogen bonds. That's what those dots represent. And so the strands on the side of them, we have the sugars and the phosphates. That forms what we call the backbone of the molecule. And then we have nitrogen base pairs on the inside. Now, we will have a whole lesson on these molecules, but just know for now that A and T always go together, adenine and thymine, and C and G always go together, cytosine and guanine. Some students like to remember that the apple's on the tree and the car is in the garage. But just know that this right here is where we find our genetic code, is in these nitrogen bases. So we see the whole nucleotide, a sugar, a phosphate, and a nitrogen base. A sugar, a phosphate, and a nitrogen base. And that essentially makes up that polymer of your nucleic acids. Now, RNA is a little bit different. Number one, it's not stuck in the nucleus like DNA. It can actually leave, and you can find it out in the cytoplasm. You can find it in ribosomes. And your RNA, there's 3 different types. We have mRNA, which serves as a messenger. We have tRNA, which transfers amino acids in protein synthesis. And we have rRNA, which is part of the structure of a ribosome. Now, RNA is different in that there's only 1 strand, and this is why it can sneak out of the nucleus, is because it's smaller. It has ribose instead of deoxyribose. And there are no T's in RNA. Instead, RNA has a base known as uracil. And we'll talk more about that at a later date. All right, so just a quick side-by-side comparison. DNA is double-stranded. It has deoxyribose, and it has T's. All right, whereas RNA is single-stranded. It has ribose as a sugar, and it has uracil. And that is going to wrap up our lesson on macromolecules. So there are 3 important videos that I included. You guys may remember the Amoeba Sisters from freshman biology. Crash Course is really more at a college level, but this particular video doesn't have nucleic acids. There's a whole separate video specifically on nucleic acids. And then Khan Academy is also another good resource. So we're going to be doing several practices over macromolecules and some little virtual labs to help you understand these in more detail. It's a lot of information. You will need to spend some time with it and make sure we know the difference between our 4 macromolecule groups. And that's going to be all for today, guys. I hope you guys are doing well. Let me know if you have any questions or concerns, and hopefully I will see you soon.